uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
PCR stands for polymerase chain reaction. This is a human-made way to replicate DNA. PCR is good for multiple reasons. For one, in forensic units, they use it to replicate DNA so they can catch a killer who only left a single hair strand behind. One problem of using PCR is that if you happen to have too much DNA and still use PCR, then it results in packed DNA in confined space of the reaction vessel, which can therefore result in false printing.
In most eukaryotes, the expressed segments of the gene, called exons, are separated by intervening sequences of nucleotides, called introns. The exons and introns are transcribed by RNA polymerase, generating what is called precursor messenger RNA. The introns are then excised from the precursor messenger RNA, and the exons linked together to form the mature messenger RNA. Prokaryotic cell DNA does not contain introns, and prokaryotic cells are therefore unable to remove the introns from eukaryotic DNA and make functional messenger RNA. Therefore, eukaryotic DNA cannot be cloned directly into prokaryotic cells to make useful eukaryotic proteins. Before eukaryotic DNA can be cloned into prokaryotic cells, the mature eukaryotic messenger RNA must be isolated and used to make DNA without introns. The enzyme reverse transcriptase is used to convert the purified eukaryotic messenger RNA into double-stranded DNA. The resulting DNA, called complementary DNA or cDNA, can then be cloned into a bacterial cell. Genes cloned as cDNA can be transcribed and translated by the bacterial cell machinery.
This video shows formation of cDNA. cDNA stands for complementary DNA. This is used to make a complementary strand of the DNA. This then clones the DNA strand. This all ties into DNA fingerprinting.
DNA fingerprinting is a method of identification based on an individual's DNA. In traditional DNA fingerprinting, the DNA is collected from cells such as a blood sample and cut into small pieces using a restriction enzyme. This generates thousands of DNA fragments of differing sizes. The fragments are then separated on the basis of size using gel electrophoresis. The separated fragments are then transferred to a nitrocellulose or nylon filter. This is called a gel blot. The DNA fragments within the blot are permanently fixed to the filter, and the DNA strands are denatured. Radio-labeled probe molecules are then added that are complementary to sequences in the genome that contain repeat sequences. These repeat sequences tend to vary in their length among different individuals and are called variable number of tandem repeat sequences, or VNTRs. The probe molecules hybridize to DNA fragments containing the repeat sequence, and excess probe molecules are washed away. The blot is then exposed to an X-ray film. Fragments of DNA that have bound the probe appear as dark bands on the film. As seen here, the samples designated 1 and 2 have a different pattern of bands, indicating that they came from 2 different individuals. Traditional DNA fingerprinting has been superseded by an automated procedure in which microsatellites, which also contain variable repeat sequences, are amplified by PCR. The small amplified DNA fragments are fluorescently labeled and subjected to high-resolution gel electrophoresis. A fluorescence detector determines the amount of DNA in each amplified fragment, thereby generating a graph that depicts the pattern of fragments in a particular individual.
DNA fingerprinting is a process using probes to examine DNA samples that have been cut by restriction endonuclease. DNA fingerprinting is good and bad. It helps us solve crimes and put away bad people. However, people have debated the issue of, is it fair or not? People may be rejected from certain healthcare if their DNA reveals something about them or even their minority. Therefore, DNA fingerprinting can be bad, but for the most part, it is good.